Hi, my name is Karolina. I'm a virology PhD student, and because I believe virology is full of fascinating stories, I decided to share some of them. Maybe you noticed media reports talking about ancient viruses found in permafrost, sometimes even using catchphrases like "zombie virus revived from the ice." And I'm not exaggerating. Even CNN used the term zombie virus in a headline. So today I'd like to talk about where these stories are coming from and what research was done on viruses or their fragments conserved in permafrost. On viruses that were discovered in permafrost samples as old as 50,000 years. On viruses that can sometimes still be infectious, even after all this time. So what exactly is permafrost and how it can conserve ancient viruses? Well, permafrost is basically frozen ground. The official definition says that it is a layer of soil or rock that remains frozen for at least 2 consecutive years, but it can be much longer. It is typically found in polar regions and in mountainous areas, and there is a lot of variability in how it can look like. Depending on the climate, permafrost can be relatively shallow and not very old, but it can also be hundreds of meters deep and hundreds of thousands years old. And as such, it presents a unique record about past climates and ecosystems, because this frozen ground can exceptionally well preserve both organic and inorganic materials. I read a quote saying that the preserving qualities of permafrost are so good that if you put a yogurt in permafrost, it may still be edible 50,000 years later. The reason permafrost has such good preserving qualities is because it is cold, dark, and there is lack of oxygen and limited microbial activities. But among the things that can be conserved in permafrost are not just archaeological artifacts or yogurts. Permafrost can also preserve organisms, and it acts as a huge reservoir of ancient microorganisms. And furthermore, it has been shown that bacteria isolated from permafrost samples can sometimes remain viable or could be revived from spores after at least tens of thousands of years. And what makes this issue even more interesting is that permafrost is vulnerable to climate change and it is thawing. In the articles I read, it was pointed out over and over again that the climate change is affecting areas with permafrost disproportionately, and regions like Siberia or Alaska are heating up faster than is the average rate of climate change across the world. The thawing of permafrost could trigger shifts in ecosystems, cause the release of greenhouse gases, and moreover, there are concerns regarding the potential risks posed by ancient pathogens infecting plants or animals that could be conserved deep under the ground. So the first question we may ask about viruses in permafrost is a general one. Is even something there? Were some ancient viruses preserved well enough that we can find traces of them deep under the surface, even after thousands of years? One way to look for these viruses is to look for their genetic information, so either for viral DNA or RNA. The first thing that has to be done is, of course, to obtain samples from deep underground. In the studies I read, this was typically done by drilling with a drilling rig, and the samples were collected from depths ranging from just centimeters of surface layers to several meters in the ground. After obtaining the sample, it is crucial to prevent contamination by today's microbes. That is why typically you don't work with the whole sample, but in a sterile environment, you cut away the outer shell of the sample and then only work with its core. I read a paper that was focusing on the study of ancient microorganisms in a glacier, And in this paper, they were testing the efficiency of this approach. They prepared a frozen sample, which they purposely contaminated by adding known bacterium, virus, and free DNA contaminants all over the surface of the sample, to test how well they are able to remove these known contaminants. So they proceeded by cutting away the outer layers of the ice core and by washing it with sterile water, and they were able to successfully remove the contaminants they introduced. So they confirmed that with this method, they can collect truly ancient samples without modern microbes. From the thawed permafrost samples can be isolated nucleic acids such as DNA. This obtained DNA does not belong to one specific organism, but it contains a mixture of the remaining genetic material from all the organic matter present in the sample. The DNA can then be sequenced, and the sequences are bioinformatically analyzed. This analysis would contain multiple steps. First, you have to check the quality of the data, and it is necessary to remove some artifacts or low-quality reads. The obtained short DNA sequences are assembled into larger contigs and compared to databases of known sequences to identify the organisms present in the sample. The sequences can also be functionally annotated, and you may predict functions of the detected genes by comparing them to databases of known protein sequences. And by these methods, were really discovered traces of viral DNA in permafrost. For example, from viruses belonging to Mollivirus family that infects amoeba. But researchers didn't just look for viruses with DNA genome. Some studies focused also on RNA viruses by looking for genes encoding their RNA polymerase. And again, they were successful and found, for example, viruses belonging to the Rhabdoviridae family, which is a large family of viruses infecting not just animals but also plants or fungi. But the most famous member of this family is the rabies virus. These traces of viruses were detected in permafrost samples as old as 50,000 years, with the age being typically determined by radiocarbon dating of the sediment layer the sample was collected from. So we know that in permafrost can be detected traces of viral DNA and RNA. But do the traces of nucleic acid represent only some remaining viral fragments, or are these samples coming from intact viruses that may still be infectious? A team of French scientists, led by Jean-Michel Claverie, tried to answer exactly this question. They worked with permafrost samples from Siberia, and they focused on viruses affecting amoeba. In their approach, they successfully used a culture of Acanthamoeba cells as a bait for these ancient viruses. They inoculated the amoeba culture with a sample obtained from permafrost, and then they looked for virus replication. It should be noted that in the growth media for the amoeba were present antibiotics to prevent bacterial contamination of the experiment and also antifungal medication. The researchers used transmission electron microscopy, and they were able to observe the infection and replication of viruses in the amoeba cells. They could detect how the viral particles enter the cell via phagocytosis. Then, after several hours, they observed changes in the Acanthamoeba host. The cells were rounding up, and they started to lose adherence to the surface. And inside the cells could be detected formation of new viral particles in so-called viral factories, which are basically structures formed by the hijacked cellular machinery where viruses replicate and assemble new viral particles. The replication cycle lasted typically about 12 hours and ended with the lysis of the host cell and release of hundreds new viruses. After cell lysis, the viral particles were purified by ultracentrifugation and further analyzed by DNA sequencing. For example, for a newly discovered pandoravirus isolate, this analysis showed that the discovered virus is similar, but still significantly different from the contemporary strains. Among the discovered viruses were viruses coming from 30,000-year-old samples, and one of the samples contained infectious particles even after it was buried under the ground for almost 50,000 years. By this method, they identified 15 different viruses that were still able to infect Acanthamoeba even after thousands of years in permafrost. There was also another study that took a different approach on how to hypothetically detect ancient replicating RNA viruses. The researchers started by slowly thawing their permafrost samples in 4 degrees Celsius and in darkness for 3 months to better represent how the permafrost thawing really looks like in the environment. They were reasoning that RNAs are much more unstable molecules than DNAs, and that by looking at the samples processed this way, their data will better represent how RNA viruses may emerge from permafrost during its thawing. And they argued that it may even potentially represent RNA that was proliferating during the thawing process. But as they admit, for such conclusion, they would also need to have a comparison of what would be isolated from these samples if they were not thawed like this, to see if there really is some distinction and some RNA viruses may be replicated during the thawing process. So their study is not conclusive, but it is still a very interesting approach. Okay, but what about pathogens? So far, these revived viruses are still only infecting single-cell amoeba. They are not human pathogens. And as the scientists who work on them say, this is intentional from their side. They chose to work with amoeba-infecting viruses because it is not too risky. And in their research papers, the authors even sort of criticize scientists that are trying to study potentially more dangerous permafrost viruses. Which is a point I am not sure I completely agree with. I think that since we have very well-secured biohazard laboratories, we should really use them to be able to evaluate the potential threat. Not studying these viruses may be considered to be the safe option, but closing our eyes will not make the threat disappear. One cautionary tale about the potential dangers of permafrost thawing came from the Yamal region in Russia. In 2016, the region experienced an anthrax epidemic that was linked with thawing permafrost. Anthrax is not a viral disease. It is caused by a bacterial infection by the bacterium Bacillus anthracis. It causes a systemic infection in animals including humans, and the course of the disease typically varies in seriousness based on the route of infection. Bacillus anthracis is able to persist in the environment for a long time. In unfavorable conditions, the bacterium forms metabolically inactive spores, which can survive for decades or even longer. These spores are not susceptible to antibiotics and they are very resistant. But when they are introduced to a new host, they begin their replication cycle again. Anthrax was previously endemic in the Yamal area, but the region was anthrax-free for more than 50 years. And because of that, was in the year 2007 discontinued the mandatory vaccination of reindeer. And this created an open space for possible reintroduction of the anthrax disease. In 2016, reindeer herders began to report they witnessed deaths of their reindeer. The previously healthy animals would become suddenly weak and unable to walk. Typically, they would have nosebleed, move sluggishly, fall, and die within several hours. Laboratory testing of samples from the affected animals then confirmed the presence of Bacillus anthracis. This outbreak killed more than 2,000 reindeer, led to hospitalization of 90 people, and unfortunately, one child died as a result of the infection. The epidemics also caused significant economical losses for people, because many of the surviving reindeer were exterminated in effort to stop the outbreak. And thousands of people were also forced to relocate, or they have been quarantined. It was suggested that the likely source of the infection was a previously buried reindeer that died of anthrax about 80 years ago. The reindeer herders reported that all the affected herds had been driven through the one same place in tundra where a thawing permafrost caused a landslide. This allowed the animals to get infected, and the spread of the infection was supported by a large number of unvaccinated reindeer, but also by a lack of experience in anthrax diagnosis. Because, like I said, anthrax haven't been seen in the area for more than 50 years. This case was very unique, but it also showed that permafrost is able to conserve human pathogens and that they may be reintroduced in the environment. But let's move back to viruses. As I've mentioned before, so far we haven't seen a revival of some ancient human infecting virus. But when we look at the genomic traces discovered deep in the ice, we can find viral DNA belonging to families, which are known to have members that also cause illness in people, for example, pox viruses or herpes viruses. Interesting discovery was made in the year 2004 in Siberian permafrost, where was discovered a gravesite with frozen mummies dating back to the 17th century. And PCR analysis showed the presence of fragments of variola virus, the virus that caused smallpox. However, these were just small DNA fragments, and luckily not infectious particles. The authors confirmed this when they failed to detect larger intact DNA segments. Besides human pathogens, problems may of course be caused also by novel viruses infecting animals or plants, which may, for example, lead to economic losses. Also, with such hypothetical viral infections, there is the disadvantage that for viruses we don't have universal treatment, similar to broad-spectrum antibiotics for bacterial infections. There is also another aspect that should be considered, and another way how the permafrost reservoir could affect us today. Even though the DNA immobilized in permafrost can come from dead or even extinct organisms, it also represents a reservoir of genes that other microbes could uptake by horizontal gene transfer, which is a common process in which microbes uptake DNA from other species. This way, these ancient genes may be reintroduced into modern organisms. And what genes could be transferred like this? For example, sequencing of permafrost samples revealed the presence of genes encoding antibiotics resistance, or for example, genes encoding bacterial virulence factors. So the question remains: should we be worried about ancient viruses? I personally look at this in two ways. On one hand, the presence of these viruses in permafrost is a fact, and I believe that we should study them to understand the risks as well as possible, especially as the climate changes and permafrost slowly thaws. But on the other hand, it is not something that would keep me up at night, because viruses are literally everywhere in the environment, and our next big potential epidemics is just as likely, or rather more likely, going to be some zoonoses spilling from animals. And we can also assume that the majority of viruses present in permafrost would not pose a threat to human health. We can, for example, expect a lot of bacteriophages or these amoeba infecting viruses. So, to conclude this story, we can briefly summarize some of the main points. Studies of samples from permafrost show the presence of viral DNA and RNA in samples frozen for as long as 50,000 years. Large portion of these detected nucleic acids likely represent only remaining fragments of viruses, but some of the viruses remain intact and infectious, which was shown by the discovery of 15 different viruses from permafrost samples that are still able to infect Acanthamoeba. So far, no human viral pathogen was revived like this, but an anthrax epidemic originated from an 80-year-old burial site showed that the introduction of conserved pathogens is possible. And while the existence of these viral reservoirs is not a reason to live in fear, I personally think that these viruses are definitely worth studying. And that's it for viruses in permafrost. Once again, thank you so much for listening all the way through. It makes me very, very happy. If this episode made you feel that zombie apocalypse may be imminent and that you maybe want to improve your karma a bit, you can give the podcast a rating or recommend it to a friend. The sources for this episode will be listed in the episode description, and the new episode should be ready in a month again. If you want to be notified, you can follow the podcast in your podcast app of choice or also on Instagram. And once again, thank you for listening.